Yeah, so I'm going to talk about the temporal dynamics of plasma phospho-tau in Alzheimer's disease. I report no disclosures. So, as you probably know, plasma phospho-tau at 3-in-1A1 has been recently proposed as an accessible, affordable, and minimally invasive biomarker for Alzheimer's disease. Despite that several previous cross-sectional studies have consistently demonstrated that plasma phospho-tau is a good marker of both amyloid and tau pathology in Alzheimer's disease, we don't have yet longitudinal studies evaluating how plasma phospho-tau actually changes over time. We therefore don't know when in the disease course plasma phospho-tau becomes dynamic or becomes abnormal. So, in order to try to reconstruct the temporal trajectory of plasma phospho-tau in Alzheimer's disease, and determine when it becomes abnormal in relation to other Alzheimer's disease biomarkers, we analyzed data from more than 1,000 active participants with available plasma phospho-tau, amyloid PET, and cerebrospinal fluid biomarkers. In 938 participants, we had longitudinal measurements of plasma phospho-tau acquired over a median follow-up time of 2.9 years. And we did not have tau PET at baseline. However, in a subset of participants who continued in ANI3, almost 300, tau PET was performed over an average of 6 years since baseline. Okay, so here we have the results. We first look at the associations between plasma phospho-tau and regional amyloid PET. So in the first row here, we can see the cross-sectional relationship between these two markers across the clinical continuum. We can see that the association was stronger for symptomatic stages and when amyloid pathology is present in widespread regions of the brain. This suggests that plasma phospho-tau might better reflect relatively advanced disease stages in terms of amyloid deposition. This observation was confirmed in the longitudinal part of the analysis. We can see how plasma phospho-tau changes accompanied longitudinal amyloid accumulation in widespread brain regions, even in cognitively normal individuals. Regarding global markers of amyloid pathology, results are pretty consistent. We can see that plasma phospho-tau becomes abnormal for relatively high burdens of amyloid pathology. However, in the longitudinal part of this analysis, we can see that the earliest dynamic changes in plasma phospho-tau could be detected even before these amyloid markers reach the abnormality threshold, suggesting that the earliest dynamic changes in plasma phospho-tau occur very, very early in the disease course. So together, these findings indicate that amyloid pathology precedes elevations in plasma phospho-tau. Now it's time for look at tau markers, in this case to cerebrospinal fluid phospho-tau. As expected, phospho-tau in blood and cerebrospinal fluid were strongly correlated, and importantly, the associations were only present in amyloid-positive individuals, suggesting that plasma phospho-tau reflects tau pathology only in Alzheimer's disease. Also important is the fact that plasma phospho-tau was predictive of tau PET signal 6 years later. Both baseline measurements and longitudinal changes were associated with widespread tau pathology in both initially impaired and initially unimpaired individuals. Again, associations were only present in amyloid-positive subjects. So this result suggests that maybe plasma phospho-tau elevations might precede neurofibrillary tangle aggregation as measured with tau PET. And finally, we employed a previously developed method for the construction of long-term temporal trajectories of biomarkers using longitudinal data. With this method, we estimated that plasma phospho-tau becomes abnormal approximately 5 years after amyloid markers. And we can also see that plasma phospho-tau follows very, very similar dynamics as compared with cerebrospinal fluid phospho-tau. So together, the results of this study can be summarized in the following sequence of events. Amyloid seems to come first, then elevations in plasma and cerebrospinal fluid phospho-tau, followed finally by neurofibrillary tangle aggregation as measured with tau PET. So that was it. I would like to thank all my colleagues at the University of Gothenburg. And if you want to see the published version of this study, you can check the January issue of Brain. Thank you very much.
Thank you. Very nice presentation, and thank you for keeping to time. As we said earlier, apologies that we can't take questions for the data blitz, but if you'd keep an eye on the Q&A, we'd be grateful. Thank you very much. So